My name's David Largaespada. I'm one of the co-founders of B-Mogen. And yesterday at a session on cell manufacturing, it was suggested that non-viral gene transfer for human cell therapy, gene and cell therapy might be 10 years away. And if you don't remember anything else about my talk, my real main message is that using DNA transposons, we can achieve non-viral gene transfer that's clinically applicable now. So at B-Mogen, we've been tackling the industry's most complex genome engineering challenges in 3 areas. So for example, in our custom gene editing service, which is a part of the company that's grown at 1,200% the last 2 years, we've earned an industry reputation for tackling complex projects and achieving them, completing them quickly. We're also the first company in the world to engineer the mitochondrial genome in human cells. This is technology that we have exclusive license to from the Mayo Clinic, and we think we're on the cusp of being able to edit and correct specific mitochondrial genetic mutations. But today I want to focus on our disruptive transposon platform that allows us to achieve non-viral gene delivery for gene and cancer immunotherapies. And this is an approach that we think can save millions of dollars in manufacturing costs, and this technology is not exclusively licensed to any one single player at present. So DNA transposons offer an attractive way to achieve permanent gene transfer and expression in human cells. And in this sort of application, a transposon vector is delivered on a plasmid DNA. The genetic cargo is flanked by sequences called inverted terminal repeats, and that plasmid DNA is delivered to cells along with a source of transposase enzyme delivered as DNA, RNA, or protein. Once in the cell, the transposase recognizes the inverted terminal repeats, excises the genetic cargo from the donor plasmid, and integrates it into the host cell chromosome, allowing permanent gene expression. So multiple transposon families exist in nature, although most have been evolutionarily silenced. Some examples are shown here. So Sleeping Beauty from the TC1 Mariner family and Piggyback from the family that's named after that. Piggyback have been used for gene therapy in humans, and these transposon systems are licensed to Merck and Poseida respectively. And at B-Mogen, we focused on another family of cut and paste DNA transposons called the HAT family, and in particular, a transposon called TC Buster. Now, one obvious application of transposons for gene delivery is to engineer human CAR T cells. And in fact, my lab and other founders of B-Mogen were part of the first in-human CAR T cell trial using transposon-based CAR delivery. And in this work, a CD19 specific CAR T cell was generated, product was generated by Sleeping Beauty gene transfer. And these CAR T cells were provided as adjuvant therapy post-hematopoietic stem cell transplantation for B-cell leukemia. And these results have been reported. And this was demonstrated to be safe and effective method for transgene introduction into T cells, and showed evidence of efficacy as well. And as I mentioned, this technology is exclusively licensed to Merck. The point of this, though, is that it shows that DNA transposons can be used in human cells safely. So there are a number of advantages that transposon gene delivery has over viral gene delivery. So this would include many things, but both methods can mediate stable gene transfer and have been used clinically. But viruses have certain cargo sequence and cargo size limitations that DNA transposons don't have. So the sequence isn't limited that we can put inside the transposon vector in any way, and we can achieve gene transfer of large cargo compared to what's achievable using retroviruses or lentiviral vectors. Retroviruses are also immunogenic, and they prefer to integrate near or within genes, raising the possibility of insertional mutagenesis. And the potential for this is much reduced using DNA transposons, which don't have those same preferences that viruses do. One big advantage of DNA transposons is that we estimate it's 2 to 5 times cheaper to manufacture cells after gene transfer using a DNA transposon compared to viruses. Now, one potential problem that we've tackled is that delivery of naked DNA to primary human cells like T cells can be toxic. However, we've overcome this using proprietary cell handling and engineering methods, as I'll show in the next slide. Now, we know that lentiviruses and retroviruses can be highly efficient for gene delivery to primary human cells. As I'll show you, we can also achieve high efficiency gene delivery and expression using hyperactive transposases. So as I mentioned, one problem with DNA transposons was DNA toxicity. And under standard gene delivery methods, compared to delivery of mRNA, for example, if you deliver DNA to T cells, it's very toxic and half the cells aren't viable. But using our handling methods, we can eliminate this toxicity. And this greatly improves the expansion that we can achieve of gene-modified T cells ex vivo. Now, we very quickly came up with some hyperactive TC buster transposases that allowed us to achieve high gene transfer frequency in primary human T cells. We have a comprehensive patent portfolio for therapeutic use of family transposons at B-Mogen. Here you can see percent GFP transgene delivery to primary human T cells with transposon vector alone or various hyperactive TC buster transposases we engineered. And we quickly developed a transposase that was equal to or better than a version of Sleeping Beauty called SP11, which has been used clinically that I mentioned before. Now, the gene transfer rate wasn't as high in this experiment as a hyperactive version of Sleeping Beauty called SP100X, which is not used clinically. And one reason that Sleeping Beauty 100X is not used clinically is that it has a high copy number per cell, which is potentially dangerous, and above that the FDA considers a safe number of integrants per cell. Whereas with the TC buster transposon system, we have a low and acceptable copy number of integrants per cell. Another advantage of TC buster compared to Sleeping Beauty is it's affected little by cargo size, unlike Sleeping Beauty. And here you can see the fold change in gene delivery from a 2 kb vector for a 3 or 5 kb vector. The Sleeping Beauty 11 or SP100X are greatly affected by the increase in size, and that's less of an effect with our TC buster transposases. So more recently, our procedures for transposon gene delivery have increased stable integration rates substantially over other methods. We can now achieve 40 to 50% stable gene transfer and expression. These are our results with high viability, and these are results from 3 different donors. And we're currently working on methods to select for the stable gene integrants so that we can produce pure populations of transgenic CAR, TCR, T cells. Another advantage of our method is that our engineered T cell populations have a high frequency of T stem cell memory cells, which have been shown by others to more efficiently engraft and expand in host and control tumor. So we get, for CD4, 60 to 70% T stem cell memory cells, and for CD8, 80 to 90% of the cells are this stem cell population. It's also worth pointing out that our non-viral gene delivery platform is compatible with simultaneous gene editing. Here you can see percent gene modification for 3 different loci and 3 different donors. So we have a lot of experience in editing primary human cells using targeted nucleases. And this includes multiplex gene knockout. So we're able to target 2 genes and knock out 2 genes with greater than 90% efficiency at the same time as we deliver the transposon. And this Western blot shows that in 3 independent donors, we can achieve near complete loss of protein expression for these 2 different loci. Now, this slide shows some of the most important data I'll show, which is successful T cell delivery and expression of therapeutic CAR and TCR constructs with TC buster. So from 4 different donors, you can see percent stable integration and expression of 2 different CARs and a TCR transgene, where we get average integration and expression rates of 25 to 30% with greater than 40-fold expansion in highly viable populations. Our TC Buster transgenic CAR T cells are highly functional tumor killers, so we see high specific killing in antigen-positive cells, as you can see here, percent killing with different effector to target ratios with three different donors, and low background killing of antigen-negative cancer cells, or mock transfected cells here. So some of the key takeaways from today's talk are that the recent advancements have improved transposon vector clinical applicability substantially. And we've developed methods to eliminate plasmid DNA toxicity. We can achieve large cargo size gene transfer. The copy number per cell is low and in an acceptable range. Our engineered T cells have a high frequency of therapeutically relevant T stem cell memory cells. And this is now an effective method for gene delivery for clinical use. So among the future near goals that we have, we're developing clinically applicable selection methods to produce pure CAR and TCR transgenic T cells. We're identifying additional hyperactive mutants and working to provide all the information required for CMC. And this platform, again, I want to emphasize, is available to partners. We've already formed roughly 10 partnerships with industry leaders, providing proof of concept for our delivery platform. And we're open, if you're interested, we're certainly open to meeting with you and talking about delivering your cargo using TC Buster. So this last slide just shows some of the key personnel at B-Mogen, our scientific advisory board and board of directors. And I will stop there, and I'm happy to take any questions you might have. Thank you.